Hi, class. Today we're going to go over regulation of gene expression. And I want you to think real quick before we begin this section the term regulate. What does it mean when someone asks you to regulate something? Or sometimes some items might have regulations. Basically, like when you regulate something, it's almost like a set of rules, a series of steps. It's like somewhat of a control factor. Okay. And when we're talking about regulation of gene expression, we're looking at control of when genes are turned on or off, right? So if we look at this illustration right here, we see that all of our cells have our signal transduction response. Remember, we have the receptors on the outside of the cells that receive signals. Okay, that signal can then be transferred to the nucleus. The nucleus will then give directions to tell the cell what it needs to do. In this instance, we could see that it's making a protein. So that means that it has to turn transcription on. So we're opening up the gene to where the specific protein is at. The gene is then transcribed into RNA. RNA gets edited and then it's transferred to the ribosome where it gets translated. And once it gets translated, it's into the protein. Okay. And then from there, it will be expressed. So let's go to take a look at the series of steps and what controls gene regulation. All right, the next couple of slides, you could read these on your own. This is what the AP exam expects you to understand. So this is what the AP exam expects you to understand. Same thing here. And we're going to go through each of these learning objectives as well. And here. All right, so let's look at prokaryotic regulation. So prokaryotic regulation, we're looking at bacterial regulation. Now, please note that we also have bacteria that live in us, okay? So bacteria are just not free-floating like found obviously on surfaces, but they're also found inside of us. And it's essential for us to have certain bacteria because it allows us to function, okay? So bacteria do not require the same enzymes all the time. The enzymes are produced only when they're needed. Okay, why would I only make things when they're needed? So think, why do I make things when they're needed? Well, I don't want to make something all of the time because it would waste energy. Okay, and if I'm an organism, I want to try and conserve as much energy as I need for days when I don't have a whole lot. Okay, so the following scientists, Jacob and Monod, in 1961, proposed the Operon model to explain regulation of gene expression in prokaryotes, the Operon model. Okay, an operon is a group of structural and regulatory genes that function as a single unit. So you got to know this term operon. Now this unit is going to be a little bit of vocab heavy. So please pay attention to the words that I'm going to tell you that you should know. Operon is one. And once again, it's a group of structural and regulatory genes. Okay, genes that function as a single unit. So they function as a single unit. When we look at bacterial metabolism, or how do bacteria break down molecules, bacteria need to respond quickly to changes in their environment. Remember that they reproduce very quickly and they respond to their environment because they're located in the environment. If they have enough of a product, they need to stop production. Why? So let's say they made everything that they need, okay? So you want to tell it to stop. So why? Because it's a waste of energy to produce more. If I already have enough of a product, I'm not going to exert more energy to make something that I already have. And how we do that is we stop the production of the enzymes for synthesis or for building. Okay. However, if they find that new food or the energy source, they need to utilize it quickly. Why? So if they find energy, they need to use it for metabolism, for growth and reproduction. And so to do that, they have to start the production of enzymes for digestion. Same thing like with us. We get hungry, we eat, we need to make enzymes for digestion. Once we've made enough enzymes in our systems as well, then our body has to also signal itself to tell it to stop. Okay. So cells vary the amount of specific enzymes by regulating gene transcription. So look at this term, regulating gene transcription. Okay. So you have to remember that transcription is always going to be DNA to RNA. So DNA to RNA, okay? And if I'm regulating it, then that means that I'm looking at the steps and seeing if everything is what it needs to be done, okay? Is it regulated? So we can turn genes on or we can turn them off. So I can make something or I can stop it. So if I turn genes off, for example, so if the bacteria has enough of this protein called tryptophan, then it doesn't need to make the enzymes used to build tryptophan. Okay, so once it has enough, I want to be able to turn the button off. Okay, now if the bacteria encounters a new sugar, like lactose, then it needs to start making the enzyme lactase to digest it. So then I want to turn something on. Okay. So I'm either going to be turning genes off or I'm going to be turning them on. And I'm going to regulate gene transcription in that manner. Right. So I'm either going to tell my body to make something or not to make something. All right, here's a brief video on the Amoeba Sisters. You can go ahead and watch that on your own. It does a really good job of going over the operons. All right, so bacteria group genes together. These are some terms that you're going to have to be familiar with as well. So these are some terms you're going to have to be familiar with as well. We have the operon, and like we said, the operon is going to be the genes that are grouped together with related functions. So these are genes that are grouped together. So the example would be all the enzymes in a metabolic pathway. So if I look here, this here is going to be our operon. Okay, these little squares are genes. Okay, and remember, genes are strands of DNA. So we have our DNA as we see here, and basically it would run like this as well, right? Okay, so then we would see our DNA, and then this area here would be this gene segment for TRPB. This here would be the DNA segment or the gene for TRPC, TRPD, and TRPE. Okay, but in science and in biology, we're going to symbolize the squares, those, as our gene blocks. All right. Then we also have what's known as a promoter region. So the promoter, when you promote something, it's a start. So RNA polymerase bind is the binding site. And if you recall, RNA polymerase builds RNA. So a single promoter controls transcription of all genes in the operon. It's transcribed as one unit and a single mRNA is made. Okay. Then we have what's known as the operator. The operator is the DNA binding site of a repressor protein. Okay, so the operator would be like your switch. Okay, so it's like a light switch. It's going to be turned on or it's going to be turned off. Okay, it's going to be turned on or it's going to be turned off. So if it's turned off, RNA polymerase cannot read the gene sequence. Okay, so that means that the gene would be turned off, right? Now, if the operon is turned on, notice that there's no block. The RNA segment can then read the DNA and can make our RNA as we need it. Okay? All right. So how can these genes be turned off? We have what's known as a repressor protein. And what it does is it binds to DNA at the operator site. It blocks RNA polymerase from reading the DNA strand and it blocks transcription. So whenever we say transcription is turned off, that means that the gene is not being read. So for example, if I'm not drinking milk, my body doesn't need to make lactase. So that means that this gene to make lactase is turned off. So there's basically here's our switch as we see. So here's our switch. Notice here that it's blocked. So RNA polymerase cannot read it. Okay. As soon as I get the signal from the outside, right? Now the signal is going to attach to this protein and then it can open and turn it on. Okay, it can open and turn it on. So a repressor protein turns something off. It turns something off. Okay. All right. So this here would be our operon model. What does this look like? So an operon, as we said, contains what's known as an operator, a promoter, and the genes they control. This serves as a model for gene regulation. Now, if I were you, I would draw this in your book so that you have an understanding of what this looks like, this illustration. You will see illustrations like this in the AP exam. So you will see illustrations like this in the AP exam, okay? So you will see illustrations like this again on the AP exam. And if you understand this illustration, then obviously it's going to help you when it's time for you to take your AP exam. All right. So like we said, here's the promoter. The promoter is going to be starting with this TATA box, okay? The TATA box is the sequence TATA because remember we're on DNA. Now, that's telling me that it's going to be the start of a gene sequence. So these are the genes that are going to be read on the DNA strand. These genes code for these specific enzymes, okay? Now, in order for these genes to be read, this repressor here, or this operator, needs to turn this switch on, okay? So as soon as it gets a signal from the outside, this repressor then is going to switch on, and then RNA polymerase can read the genes and then make, obviously, our enzymes, okay? But as long as there's a repressor here, the gene can't be read. The gene can't be read. And as we see, the repressor protein turns off the gene by blocking RNA polymerase binding site, okay? All right, there's two types of operons that we're going to look at. One is called a repressible and one is called an inducible, okay? So let's look at this first one, a repressible operon. For the repressible operon, it's going to be known as the trip operon. Trip operon because it's going to make the protein called tryptophan. So we call it the trip operon, okay? The synthesis pathway modeled. So when excess tryptophan is present, it binds to trip. Repressor protein, and it triggers the repressor to bind to DNA. Basically, what it does is it blocks transcription. It blocks transcription. Okay, so it's going to turn it off. It's going to turn it off. So, for example, once again, when I have excess tryptophan, so when my bacteria has made too much, right? It's made enough. Now I want to be able to shut it down. So this repressor protein here, or this repressor, will shut off, okay? As soon as this shuts off, RNA polymerase cannot read the genes and it's no longer able to make tryptophan, okay? It's no longer to make tryptophan. So this conformational changes the repressor and turns it off, okay? So once again, the gene is turned on, okay? So it's making tryptophan until it's made enough. Once it makes enough, then as we see, this tryptophan here is going to attach to the repressor and it's going to go ahead and turn it off. All right, so what happens when tryptophan is present, as we see here? So if tryptophan is present, you don't need to make tryptophan building enzymes. So it gets shut down. It gets shut down. All right, so once again, this is what the trip operon looks like. We have the regulator gene. Here's the promoter gene. Here's the operator. Remember, the operator is the switch. When the repressor binds to the operator, transcription is prevented. So when this active repressor here, when this is attached, it's the signal to turn it off. And RNA polymerase cannot read the genes. And then if it can't read the genes, it can't make the protein. All right, so here's our example. So when tryptophan is absent, the gene is going to be read. So the enzymes need to be synthesized, tryptophans are produced. So as we can see here, RNA polymerase is here. There is no repressor, so the operator is turned on. And as we see, it's making mRNA. mRNA then is allowed to make these specific enzymes. However, once it makes enough tryptophan, now what I want to do is I want to be able to shut it off. I want to be able to shut it down. So to do that, tryptophan then is going to attach to the repressor, and then it's going to turn off my operator. Once it turns it off, RNA polymerase cannot read the gene, okay? And it can't make the mRNA. If it can't make the mRNA, it's not going to make the enzymes. Right, the other type of operon we have is called the inducible operon. The inducible operon. Example is going to be lactose. This is found in our digestive pathway model. So when lactose is present, so I drink milk, now it's going to bind to what's called the lac repressor protein. And it's going to trigger the repressor to release DNA, and it's going to induce transcription. So what that means is it's going to turn the gene on to read the lactase genes, okay? And it's going to turn transcription on. So once again, lactose is going to attach to the repressor. Once it attaches to the repressor, then this here is going to turn on. Then RNA polymerase can read the genes to make lactase. Same thing, it's controlled. Our body's not making lactase if it doesn't need to. It's only going to make it when I drink milk. If I'm never drinking milk or I have no lactose present, this gene's going to stay shut off because there's no reason for me to make an enzyme that's not present in my system where there's no substrate. All right, it's the same thing. Here's our lactose operon. What happens when lactose is present? We need to make the lactose digesting enzymes. We need to make the lactose digesting enzymes, as we see here. Okay. All right, so if anything, this is what you should know. In a repressible operon, it usually functions in anabolic pathways, synthesizing end products. So when the end product is present in excess, the cell allocates resources to other uses. So this is when we are building, okay? The inducible operon usually functions in catabolic pathways, in digesting nutrients to simpler molecules. So this is used to break things apart. So it produces enzymes only when the nutrient is available. Okay, only when it's available. So the cell avoids making proteins that have nothing to do. Cell allocates the resources to other uses. So once again, your repressible and your inducible. So this here is going to be our trip operon, and then the inducible will be the lac operon. Okay, turning off and then turning on. All right, here's same thing. Here's another, it's repetition. So the lac operon, like we said, the regulator codes for the repressor. If lactose is absent, the repressor attaches to the operator and the expression is normally off. So I'm not drinking milk. I don't have any lactose, so my gene is turned off. When lactose is present, it's going to combine with the repressor, and then it renders it unable to bind to the operator. RNA polymerase then binds to the promoter, and the enzymes necessary for lactose catabolism are produced, meaning we make lactase. Same thing, here's our illustration when it's turned off and when it's turned on. And this is what our operon normally looks like until we drink milk. Well, that would be the signal, right? And then it's going to do something. And in the end, it's going to make a protein. All right, here's a really cool video on turning genes on and off. Please watch this because it's going to talk about how the environment plays an effect in turning specific genes on and off, especially in animals that live in different seasons to which they have to change or they have to camouflage. So please watch this because the AP exam likes these types of examples. So if you listen to it, it should obviously, hopefully stick in your mind, okay? All right, let's look at eukaryotic regulation. So such as us. Now, when we talked about prokaryotic regulation, so we looked at the prokaryotes. And like we said, if we look at lactose, prokaryotes, they have to digest sugar as well. And they have to digest milk. Some of them live inside of our gut. But when we look at eukaryotic regulation, we're looking at all of our body cells regulations. And the difference is that, remember, our eukaryotes have a nucleus. Prokaryotes don't. So their DNA, as we've seen, is just free floating, okay? Eukaryotic organisms, such as us, we can halt replication, transcription, and translation at different points, okay? So there's a variety of mechanism when we look at control of when we turn our genes on and off. So there's 5 primary levels of control. Nuclear levels, meaning that these occur in the nucleus. So we control the chromatin structure. Please don't forget, chromatin is when DNA is wrapped around proteins called histones. Okay. All right. And it helps to condense the DNA. Transcriptional control is going to be DNA to RNA. And then post-transcriptional control is going to be editing the introns and the exons, right? When we look at cytoplasmic levels, these occur in the cytoplasm, not inside of the nucleus, and that would be where translation occurs. So that's going to be RNA to the protein, and then post-translational control, basically what happens with the protein after. So there's certain steps that can be halted in all of these. All right, so the big questions is, how are genes turned on and off in eukaryotes? And how do cells with the same genes differentiate to perform completely different specialized functions? One of the biggest examples I can show you here is going to be embryonic development. So if I look at fertilization here, when the egg and sperm unite, remember that the nuclei of the the nuclei from the sperm and the nuclei from the egg are going to merge together. Okay. They're going to merge together, and then the chromosomes from each cell are going to unite, and then they're going to form you. Now, those chromosomes obviously have to have their genes turned on because they have to make a whole lot of things. Basically, they have to make a human, right? So, as we look at fetal development here at 9 weeks, I think it's 8 weeks. I can't see. Well, I'm sorry, my little deal is right here. Maybe it's 6 weeks, 14 weeks, and then 20 weeks. Notice how the embryo has changed over time. So that means that the cells, or basically the genes, are at controlled levels. Okay, so as we see here, fingers are not formed yet. As we see here, fingers are formed. So what has happened in development? What did the genes do that enabled these cells to die off to form the fingers? Okay. All right. So once again, these are the points of control. The control of gene expression can occur at any step in the pathway from gene to functional protein. So every single step here is controlled by our body. If something is not made right, guess what? Our body has the ability to shut it down. Okay. If we don't shut things down that are not made properly, as again, we can form cancer, right? Okay. So packing and unpacking the DNA is going to be our step one, and that involves DNA methylation and histone acetylation. So we're going to talk about methylation in a second, and then histones, like we said, is when the DNA wraps around the proteins. All right, we can control transcription. We can control mRNA processing. Remember, editing and the introns and the exons. mRNA transport, adding the tail and the cap, transporting it to the cytoplasm where it's going to attach to the ribosome. We can control translation, protein processing, and then protein degradation. Okay, let's look at chromatin structure. So eukaryotic DNA is associated with histone proteins. Together, they make up chromatin. So whenever you see the term chromatin, it's DNA wrapped around histone proteins. We have what are known as nucleosomes. And nucleosomes basically is DNA, which is wound up around groups of 8 molecules of histone proteins. It looks like beads on a string, and each bead is called a nucleosome. The levels of chromatin packing are determined by the degree of nucleosome coiling. Let's take a look at that. So, DNA packing is gene control. So, the degree of packing of DNA regulates transcription. If the DNA is tightly packed, right, it's tightly around the histones, the gene cannot be read. So, in order for it to be read, this DNA and these histones, it has to basically loosen up. So, once it loosens, it allows for the gene to be read. It's tightly wrapped around the histones, as we said. So if it's too tight, there's no transcription and the gene is turned off. Okay, so our gene is turned off. As we see in this illustration, we have heterochromatin and euchromatin. So heterochromatin is going to be darker, tightly packed. So as we see here, this DNA is tightly packed, so it's not being read. Whereas this area here, euchromatin, is loosely packed. So that means that these genes are being read. So here's our other illustration. As we see, our heterochromatin and our euchromatin. So euchromatin is the loosely packed DNA. Heterochromatin is the tightly packed. Okay. All right, euchromatin is loosely coiled DNA, and it's transcriptionally active, meaning that since it's loose, the DNA is being read or the gene is being read. If it's tight, okay, transcription is inactive and the DNA is not being read, so there is no DNA to RNA. All right, we see this occur with our Barr bodies. So females have two X chromosomes, but you guys only have one that's active. The other X chromosome is tightly packed along its entire length and is inactive. The inactive X chromosome is called a Barr body. Now, this only occurs in females. Why? Because if you're a male, you have XY. So that means that your X chromosome is turned on and your Y chromosome is turned on. However, for females, because we have two X chromosomes, it's redundant. We need two for us to become female. If you only have one, which is kind of silly, right? If you have one and you're XO, you have Turner syndromes, and you'd have a genetic disorder. However, we have two, and one randomly shuts off. You need two in order for one of them to function. Okay. So if we look here, X inactivation in mammalian females, we can see here this female cat is a calico cat. Okay, so calico cats are only found in females because they can exhibit this codominant allele of orange and black. So male cats, because they only have one X chromosome, they're either orange or black. Females can be both orange and black. Okay. So like we said, females have two X chromosomes. One X chromosome is inactivated in each cell, which is one by chance. So as we see here, this is an active X chromosome for the orange. This one here is inactive orange, but the black chromosome here is located or turned on. Okay. All right, DNA methylation. So methylation of DNA blocks transcription factors. So there's no transcription. So that means that we turn the genes off. And the attachment of a methyl group. So when this methyl group here is attached to cytosine, which is our nucleotide, right, or our nitrogen base, basically what it does is it's nearly permanent inactivation of the gene. So whenever a gene is methylated, the gene is turned off. Okay, so methylation means that we add this CH3 molecule to cytosine, okay, to our nitrogen base. So if a gene is methylated, as we would say, there is no transcription. Okay, so that means that transcription is blocked. No DNA to RNA. It has a big factor in what we look at in what is known as epigenetics. Okay, epigenetics is a very new up and coming discovery that we found in science, and we're looking at how environmental factors play a role in fetal development in terms of turning genes on and off. So whatever the mom is experiencing during her pregnancy can actually affect which genes can be turned on or off. So I invite you guys to please watch these videos. They're really, really, really good, and they're very informative in terms of talking about epigenetics. All right. Okay, let's look at transcriptional control now. So we're still part of regulating, and what we're looking at is regulating transcription, so DNA to RNA. So transcription is controlled by proteins called transcription factors. Okay, so these bind to our enhancer DNA. The enhancer DNA is a region of DNA where factors that regulate transcription can also bind. So transcription factors are always present in the cell, but most likely have to be activated before they bind to the DNA. So that would be like our promoter region, is what we were talking about. So if I look here, here's our eukaryotic transcription factors. Here's our enhancer region. This means that we're going to come up to a gene sequence. Here's my promoters sequence. So the promoter is the start of the gene sequence. Then I would have my switch. And as I see here would be my gene of interest. Okay. Our transcription activator here is when the signal is received, the enhancer region, then will submit these specific proteins, which will then turn our RNA transcription on. When RNA polymerase is turned, when the gene is turned on, then RNA polymerase is able to read the DNA segment and transcription is turned on high. Whenever you see on an exam or an exam question, transcription is turned on high, that means that it is DNA to RNA. That means that the gene is being read, okay? So transcription initiation. These are the control regions that we see on DNA. So first there's a control at the promoter region, the nearby control sequence of DNA, basically binding of RNA polymerase and transcription factors. This is going to be the base rate of transcription, so where it's going to be turned on or off. Then we have the enhancer region as we see here. That's the distant control sequence on DNA. It's binding by an activator protein and an enhanced rate or high level of transcription. So when this here is turned on, it's going to submit our proteins to turn on transcription as we see here. So this here will activate. It will then, as we can see here, pinch and bind to this little green molecule here, which then turns RNA or our RNA sequence to be made. All right. We can also regulate post-transcriptional control. So post-transcriptional control occurs after RNA has been made. So it operates on the primary mRNA transcript. So given a specific primary transcript, so excision of introns can vary, so meaning taking the introns out or putting them together. Splicing of exons can vary, so cutting the exons. You may accidentally cut too much into the DNA segment, which is a coding region, which can mess up. And that determines the type of mature transcript that leaves the nucleus. It may also control the speed of mRNA transport from the nucleus to the cytoplasm. It'll affect the number of transcripts arriving at rough ER and therefore the amount of gene product released per unit time. So if you think about it, the longer the strand is, the longer it's going to take for us to splice out the exons and put the introns together. The shorter the strand is, the easier it is for us to take out the exons and put the introns together. And that's going to help vary the time. All right, so as we see here, here's our alternate splicing. So we're splicing different areas in or out. All right, once that leaves, then we can control translation. What can be controlled at translation? So it determines the degree to which mRNA is translated into a protein. So it features of the mRNA affect whether translation occurs and how long the mRNA remains active. So the presence of the 5' cap and the poly end tail can control that. We have what are known as microRNAs or miRNAs. And what they do is they regulate translation by causing the destruction of mRNA before they can be translated. So this here destroys mRNA before it can be translated. So it's a specific type of enzyme. Okay. We have post-translational control. And what this does is it affects the activity of a protein product. And then same thing, post-translational control is accomplished by regulating. You're either going to turn the protein on where it's going to be sent out, or we can degrade it, meaning like we can break it down. Okay, here's an example of gene regulation using the evolution of the stickleback fish. Please, once again, watch this video as it does a really good job of giving you a visualization of transcription, translation, and control of these specific genes. Okay. All right, finally, we're going to talk about gene mutations. Okay, we've talked about these with our cancer when we learned about mitosis and meiosis. So let's just kind of briefly summarize them once again. So a mutation is a change in the DNA. The effects of the gene mutation can range from no effect to complete inactivation of the protein, okay? If it's a germline mutation, it occurs in the sex cells, meaning that these can be passed on. So any type of mutation that occurs in your germline can then be passed on into our sex cells. If it's somatic cell mutation, then the somatic cells are your body cells, okay? And these mutations you don't pass on to your offspring. These are only occurring in your own specific body cells. All right, there's two types of mutations you got to know about: point mutations and frame shift mutations. Once again, we've seen these before. So a point mutation involves a change in one DNA segment. So if I change one section of DNA or one letter, it changes the protein. So a change of one codon to a different codon. It affects on the protein vary. It can cause it to become non-functioning. It can reduce the functionality, or you can have an unaffected protein sequence. Okay. An example of a point mutation is going to be sickle cell anemia. Sickle cell anemia occurs because there's one change in the DNA segment. If I change the DNA, I can change then the protein or the amino acid. If I change the amino acid, I can change the protein. If I change the protein, remember we said it may not work or it may change shapes. Okay. Another mutation is a frame shift mutation. So we look at frame shift mutations. This is when I add or I take away a nucleotide. So I'm either inserting or I'm deleting a DNA. So the protein is always rendered non-functional. So it's never going to work properly. Why? Because I've changed the whole reading frame. So if I look at this normal, let's say this is our DNA sequence and it would be read, the cat ate the rat. Okay. Now notice here after I deletion, so I delete the C. So now I can no longer read the cat ate the rat. Notice now that the reading frame has been changed. Same thing, if I add an extra letter, it is no longer the cat ate the rat. All of these codons then are going to change the amino acid, which will always not be well. It's not a good mutation to have. Same thing, here's our point mutation in hemoglobin, as I was talking about. This is from one letter change. If I change one base, I change the protein or the amino acid. I change the amino acid, I then change the protein, and I change the shape of how the red blood cells function and work, or their structure. All right, same thing. Here's a small video on mutations. Hopefully you watch that in case you need to be clarified anymore. When we look at gene mutations, so gene mutations are segments on the DNA that can have some mutations. Same thing, these can then develop into cancer. We have proto-oncogenes, which stimulate cell division. We have tumor suppressor genes, which stop cell division. And then we have mutations in oncogenes and tumor suppressor genes can then lead and stimulate the cell cycle uncontrollably, which can then lead to cancer formation. Okay. So as we see here in this cell signaling pathway that stimulates the oncogene, this protein is going to be permanently stuck onto the signal, which is going to send signals to the cell to continuously make more and more and more cells. So this is the oncogene has been turned on. And as we can see over time, this cell is just going to form a tumor. All right. Here's another video. Same thing. This is one of our last videos for you guys to watch on mutations. This mutation has to do with the rock pocket mouse and how a gene segment can change the coat coloration of these specific mice, which can then lead to some environmental changes how the environment helps to influence gene changes. Okay. All right, everyone. I hope you all learned something. If you have any questions, obviously reach out to me on email. You guys have a great day.